Hello students, this is Dr. Faizan Mirza. In this video, I'm covering with you how the structure of collagen, just recalling the structure of collagen with you so you are able to attempt descriptive questions from that as well as the questions that come from MCQs, which often they ask because collagen is one of their favorite topics when it comes to biological molecules. So let's discuss collagen. Let's recall what collagen is. So collagen, we know it's a fibrous protein. We know collagen molecule is made up of a polypeptide and not one polypeptide. Rather, it's made up of three different polypeptides. So there are three different polypeptides that this structure is actually made up of. So this is one polypeptide of collagen. So three polypeptides, they turn around one another. So when they twist around one another, they form a collagen molecule. So this one is a collagen polypeptide, a single polypeptide, and the three polypeptides turn together to form a helix, not an alpha helix, but a helix with staggered ends. So this is called a collagen molecule. This collagen molecule, since it's having three polypeptides twisted around one another, so this follows a quaternary structure because quaternary structure is where there are more than one polypeptide turned around each other to form a three-dimensional structure. In a single polypeptide, every third amino acid is glycine. You can recall glycine is a small amino acid with hydrogen in its R group. This allow the polypeptides to fold in a compact way with one another. The staggered end also allow more strength and easy fitting of the polypeptide chain one on top of the other, so they can actually provide more strength. The collagen fibers are then made up of many collagen molecules lying parallel to one another, having covalent cross-links between the R groups of amino acid near the end of the polypeptide, as well as these glycines. They also form the cross-links with one another. So this is how the fibers of your collagen looks like. Now comes the question. The question actually came in October, November 22, region 22, are pretty recent questions. Fibroblasts are one of the cells that type of connective tissue. The cells synthesize and secrete collagen, which forms part of the supporting external cellular environment known as the extracellular matrix, which is present outside the cell. Figure 5.1 shows the primary structure of the section of a polypeptide chain of a collagen. So you can see gly means glycine. So a lot of glycine molecules are there, amino acids, including other amino acids as well. So this is first, second, third, and fourth. So every third amino acid apparently is glycine. So this looks like collagen. Explain how a primary structure is shown in figure 1, 5.1. What's primary structure? It's the sequence in which the amino acids are arranged or the order in which the amino acids are placed in the polypeptide, and these are all the peptide bonds holding adjacent amino acids together. Figure 5.1 indicates the structure of the polypeptide is suited for the component of collagen molecule. Why? Because glycine is repeated in every third amino acid. Glycine we know is a small amino acid. The R group is hydrogen, allow chain of collagen to fit more compact with one another. You need to recall some description from your chapter one of cell structure. After final processing of Golgi body, collagen is released to the outer surface of the cell by the process of exocytosis. Complete the passage to describe the process of exocytosis. And we know after the processing of Golgi body, it's packaged into a vesicle. It moves across the cytoplasm, moves along the microtubules, and they move towards the cytoplasm. It uses ATP. The phospholipids of vesicles fuse with the phospholipids of the cell membrane, and the contents are released outside. Hydrolytic enzyme known as collagenase. Hydrolytic means they are breaking enzymes, so they break collagen. Now secreted in an inactive form, cells also secrete collagen inhibitors. The activity of the enzyme and inhibitor is also regulated, so the development of the materials of the extracellular matrix is controlled. State and explain what outcome will be of composition of the extracellular matrix if collagen is inhibitor activity is needed. So in case what do we get from this information? Increase of collagenase activity will increase the collagen breakdown. When more collagen is broken down or being hydrolyzed, there will be a less stiff or less strong matrix. If collagenase inhibitor is increased, on the other hand, it will allow less collagen breakdown and more strong matrix to be made. So reading of this question, what the answer can be: increase in the collagenase inhibitor activity will prevent collagenase from binding to collagen at the active site. Less collagen will be hydrolyzed because it will not fit into the active site anymore, and the matrix will have more collagen in turn. Synthetic inhibitors have been trialed and as potential treatment for diseases caused by lack of regulation of collagen activity. Research involved investigation of mechanism of an inhibitor to state the effect of non-competitive inhibitor. and on the Km and the Vmax. So we have learned this from the chapter of enzymes that if it's a non-competitive inhibitor, the Vmax changes and it reduces, whereas the Km stays the same. If it was a competitive inhibitor, we know that in that case, the Km changes and the Vmax stays the same. So just recall that and restate that. Let's move to another question. Now, this question came in Feb March 2023, variant 22, again of collagen. Collagen is the most common structural protein in vertebrates. Collagen provides the skin with flexibility and strength. Explain how the structure of collagen fiber provides skin with the strength. Collagen fiber. They are asking about the collagen fiber. So this is the collagen fiber. In many polypeptides, they are cross-linked with one another, and many collagen chains actually are crossing with one another. So in collagen fibers, collagen molecule lab parallel to one another, covalent crossing form between these. Covalent bonds are strong. They always, whenever you mention covalent bond, mention they are strong bonds. They help in providing strength to the skin. Individual collagen molecules, they have staggered ends that give strength to collagen fibers arranged in layers along with different angles to provide support in different directions. So to provide three-dimensional support. The enzyme collagenase breaks down collagen. Collagenase has several important medical uses, such as treatment of burns of skin, treatment of burnt skin. The scientists investigate the effect of pH and the activity of collagen at 37 degrees Celsius. So you can see how the optimum temperature, how the percentage maximum activity is there on the y-axis. The x-axis shows to you the pH is the optimum pH is slightly in this range, and this is the whole range in which this enzyme works. Explained by the activity of collagen is lower at pH 8.0 than at the optimum pH. So what happens when the pH changes? What happens in the enzyme? Exchange changes the pH. We know it denatures the enzyme at pH 8. The ionic bonds holding the 3D structure, they start to break down. The active site shape changes. Less collagen fits with the enzyme to form ES complex. We know that if we are changing a lot of the temperature, then the hydrogen bonds break. So the three-dimensional shape changes. If we are changing the pH, then the ionic bonds break. Although the hydrogen bonds break as well, but the ionic bonds are the main ones which actually change upon changing pH. That's it from my side. I hope that was helpful for you. Thank you so much.